important. No, you can just catch up. It's okay. Anyway, so the IEEE brain community acts as kind of a clearinghouse and a unifying focus for all of the various IEEE societies who have brain-related research activities ongoing. And the IEEE brain community, we publish papers from the brain community. We have meetings, for example, like this one. We work with standards and roadmaps for neuroengineering. One of the largest efforts ongoing in the brain community is a neuroethics effort that's we'll hear about, I think, tomorrow. So the brain community is a very active community. It's been going on about 6 years. And if you're not part of the IEEE brain community, if you're not part of the IEEE, you should join the IEEE. And if you're in the IEEE or about to join the IEEE, you should join the brain community. You're allowed to join the brain community basically just as part of belonging to the IEEE. So I assume that everybody in this room is either a neuroscientist or interested in neuroscience or a neurotechnology person or neuroscience adjacent. You all think the brain is interesting and cool. And so you should all be in the brain community. This specific meeting was sponsored by the brain community, the EMBS, and the Ricky Society, which is Solid State Circuits. There you go. Oh, there's Ricky. Solid State Circuits. See, there's so many societies, I can't keep them straight. And the society was mostly, the conference was largely organized by Gert and Ricky, but I do want to acknowledge the most important person who helped organize this meeting, who is Cynthia Weber at the very back of the room here. Cynthia, I am just the figurehead for I'm the chair of the IEEE brain community, but like all professors, I don't actually do anything useful. All the real work is Cynthia. She runs the whole brain community, and she was largely responsible for organizing this meeting. And we also had a lot of great help from people in San Diego to organize this meeting locally. Okay, so our first speaker today doesn't need an introduction, but I'm going to introduce him anyway. It's Shankar Subramanium from here. He's a distinguished professor of bioengineering, computer science, engineering, cellular molecular medicine, nanoengineering. He's the Joan and Irwin Jacobs Endowed Chair in Bioengineering and Systems Biology. He was founding director of the Bioinformatics and Systems Biology Interdisciplinary Program. He's a distinguished scientist at the San Diego Supercomputer Center. Probably the only like neuroscience person who has that distinguishing mark. He's a fellow of the American Institute for Medical and Biological Engineering, the American Association for the Advancement of Science, and is a recipient of the UCSD Chancellor's Award for Research Excellence, and a Genome All-Star, Smithsonian Foundation and Laboratory Automation Awards winner. And he long ago received his PhD from the Indian Institute of Technology. So Shankar is a very distinguished speaker. It was very generous because he just flew in this morning from Washington, DC. I'm sure he's quite sleep deprived and grouchy. So let's all give him a big welcome. Thank you, Shankar.
Thank you so much, Jack. Am I too loud? Maybe I should bring it down. I would like to thank Gert and Cynthia, and of course, Metin, for this kind invitation. It's such a privilege to be talking here to such a distinguished audience here. Thank you very much. I should point out before I begin, I was past president of EMBS, IEEE EMBS, and I told myself that who can be the very best president? Nobody better than Metin. Metin has been like this battery which goes around saying everlasting forever. And so Metin has been really phenomenal. Metin, your leadership has really made a huge difference. And for the Brain Society, it's been really good. I want to congratulate Metin. So I would like to, I've been instructed, and I always follow instructions. I've been told that I should stand still in this space and not move more than a few inches so that people elsewhere can see me. So I'm going to try my best. My tendency is to be chaotic and highly entropic, but I'm going to desist and stay here, physically rooted to the spot. Okay, and if I move, please warn me. And so thank you for. So I would like to talk to you today about very interesting work interesting to us work in my lab, which deals with Alzheimer's disease. For the past 4 or 5 years, we've been very excited about AD. And so, I mean, as you all know, it's, one of the most debilitating disorders, and it's one of the key neurological, neurodegenerative disorders in human life today. I'll give you some statistics to illustrate what I'm saying, but this is really the reason why I want to talk, to share with you our excitement. What I would value, and I always start my talk with this, I would value extremely is any input, suggestions you have for us, because I strongly believe that we are still students in this area. And so any suggestions, advice you have would be greatly appreciated. So this work began because we, at one point in time, we learned that there is no cure for AD, and there's no cure for AD even today. There's only one approved drug which doesn't work. And so therefore, that's where we are at this point in time. So let me go on to tell you a little bit about where we are. Before we do anything else, I should do my customary thing of, how do I move this? Doesn't seem to move to the next. There it is. So before I do anything else, I should acknowledge grant funding. So my lab is funded for this particular area of neuropathologies. We were funded till recently by a Science and Technology Center grant. Jack was in fact a part of that. And then we have had a grant from Chen Foundation. We have a National Institute of Health grant, and Joan and Erwin Jacobs Endowment. And I have a National Institute of Aging grant currently in consideration. So most of you know at least a Wikipedia type knowledge of what Alzheimer's disease is. It's essentially, this is a healthy brain state. Alzheimer's disease is a shrunken brain. This is a very high level cartoon of essentially they're dying neurons with because of plaques and tangles. I'll tell you more in detail about these in a few minutes. And then there are healthy neurons mixed with those. And as a function of time, we lose healthy neurons. And this is a cartoon by somebody in my lab, Andrew Caldwell. In fact, what this really illustrates is healthy neurons illustrated by neuron-like shape and non-neurons, which is what I'm going to show you at the end of the talk, is exactly what happens in the human brain. So I'm going to start with the lesson in geography. So if you look at this map of the world, there are all these different points and color coded in some different ways. So you can ask me, what does it mean? So I want to focus on this one particular spot, which is a region in Italy, which is a red spot here that you can see. It's a region called Catanzaro, and its particular area is called Girifalco. And in 1904, they identified a patient they declared the patient as crazy or as being forgetful of everything. And then there's a hospital recording. Then they went back. This particular place has, in fact, I'll show you in the next slide. This particular place is a hospital which records, it's only hospital in Italy, which has 400 years of mental health record. And so they had 400 years of mental health record, which said that this particular patient's family heredity, if you look back many years, also had very different, they recorded it as, crazy or lunatic or, various kinds of things. And these records are still available. In fact, they said this is 1 year before Alois Alzheimer identified this as a brain disorder which is named after him as Alzheimer's disease. And so this is the family history. In fact, going back, the same family I told you about, this particular individual who was, it's a woman, was traced to go back, and in fact, all the way back for many generations, many people in that family were afflicted with Alzheimer's disease. So you can ask the question, why is this the case? In fact, let's go back to the map for a second. If you go back to the map, this person who was afflicted here, in fact, has a mutation in one single protein in their physiology, one single gene, which is called the presenilin 1. And this presenilin 1, this mutation is methionine 146 to a leucine. And this one single mutation made this person crazy. What's most interesting and exciting is that you can see the same red dot near Argentina in Buenos Aires in Argentina and in Australia. And it turns out that they had once they identified it, they found out that the genetic origin of this was really here in Girifalco in Italy, and these families came from there. They didn't need to do genome sequencing to trace this back. They traced this back from simply knowing that mutation. In fact, all of these mutations, founder mutations, are, you can see, for example, blue. You have this blue in different parts of Europe, you have in Canada, you have in US. They all have a founder mutation, and that's H163, histidine 163 to an arginine. And so this is really the map of, so this gives you a glimpse of a single mutation. Now, why am I telling you this? I'm telling you this because Alzheimer's is the 6th leading cause of death in the US. 5.8 million, this is from the Alzheimer's Association org, NIH maintained organization. 5.8 million Americans are living with AD. 1 in 10 people age 65 and older have AD. In 2020, AD-related dementia costs $305 billion. By 2050, AD is projected to cost $1.1 trillion of taxpayer money. Older African Americans are twice as likely to have AD than Caucasians, and more women have AD than men. I didn't add that here. Here they are. 2/3 of Americans with Alzheimer's are women. And after investments of tens of billions of dollars in research, there's no cure for AD. There's really no cure. And we don't even diagnose AD till the time it is too late. And so I'm going to tell you why, all of this is happening in physiology. So this is, in fact, there are 2 types of AD. One is called familial Alzheimer's disease. It's called sporadic Alzheimer's disease. All those people who had mutations that I talked to you about to come under this category called familial Alzheimer's disease. It's genetic in origin with three different proteins or genes established. Now they think there are more, but the 3 are, presenilin 1, presenilin 2, and APP. I'll tell you more about them in just a second. And then in the sporadic AD, there are many genetic variants. And this happens at a later age in life, greater than 65 years. And this happens in less than 65 years, sometimes as early as 35 years of age. I'm going to tell you how common they are, how different they are in just a second. This is these mutations that I'm talking to you about, presenilin 1 and presenilin 2, are coming from this complex, which is called the gamma secretase complex. And it's got 4 different proteins. And mutations in these different spots, I don't think you can read it very clearly. I don't want you to use a microscope to read it, but these mutations in red, which happen here, are all AD causing mutations to different degrees. The question is age of onset is different. The longevity, how long a patient lives is different, etc. Severity of the disease, but they are very penetrant. Most of them are 100% penetrant, which means if a person has this mutation, it is not if the person will get AD, it's a question of when the person will get Alzheimer's disease. So in fact, this is really very important. So if I go to the next slide, the age of onset, presenilin 1, which I told you about, the age of onset is around 43 years, mean age of onset can be as early as 35. And life expectancy is not very high. In fact, you can see that how long they live. Presenilin 2 is a bit better. APPs are somewhere in between and so forth. So this is just statistics and data. So this is very useful for people to know about lifestyle maintenance and so forth. So let's look at Alzheimer's in the patient. In fact, till about 10 years ago, the belief was the following, which is that all of Alzheimer's disease is formed with what is called plaques, which means there are these small peptides which kind of aggregate to take over the brain, which form plaques. And these plaques are really what are causing Alzheimer's disease. This was the canonical opinion. And so all dollars that were invested in research in AD focused on dissolving these plaques. That was a central theme of all research. NIH alone invested $3 billion in terms of trying to find, cures for AD. I'm going to tell you a small story about this before I go into telling you more about the details. This is called the Eli Lilly story. Eli Lilly spent 3 decades and more than $3 billion in AD therapeutics. They are the company which really have invested tremendous amounts. In fact, very early in 2012, New England Journal of Medicine published a phase 3. They were in phase 3 trials. They said Eli Lilly stops the trial of semaglutide, which is really the drug that was developed by Lilly. And New York Times said Lilly stops Alzheimer's drug trials. And subsequently, all the drug trials by all companies focused on these plaques were all stopped. In fact, Alzheimer's failure raises questions about disease-modifying strategies. And the ultimate journal, Forbes, said that the AD hypothesis is dead. The hypothesis of plaques is completely dead. So therefore, in fact, this research is more or less stopped in terms of focusing only on AD therapeutics. So why are Alzheimer's drug discoveries a failure? Why is it that we cannot do this? So this brings us to the question of this entire talk today. There are lack of model systems to experiment. You cannot induce AD in mice. Mice don't show AD. Even primates don't show AD. So there are no really good model systems. We cannot go into humans and invasively look at whether there's AD, Alzheimer's disease or not. You cannot do that. So therefore, there's no easy way to diagnose AD or to treat AD. We are attacking the wrong mechanisms, which is what I told you about using plaques. And so therefore, doesn't address the disease. There's no screening strategy. There's no good screening strategy because if you don't have humans, how are you going to screen in non-humans? In animal primates, animal models don't show AD. So how are you going to screen and test? And the thing that, and there are no strategies for early detection. Blood, there's a blood-brain barrier. Blood doesn't show really all of the key, any markers that can associate strongly with Alzheimer's disease. So what are the solutions? There are engineering solutions to this problem, which is what I'm going to focus on talking about today. First is making personalized brain organoids. With the advent of stem cell technology today, it is possible to make brain organoids. And these are really personalized, precision medicine context where we can make brain organoids in single humans. Colleagues of mine in UCSD have spearheaded this effort, and there are really excellent brain organoids that people are making today. My own lab now is working very heavily on making brain organoids with Alzheimer's patient samples. Second, there's developing a drug chemical reactor screening workflow. We need a screening workflow. We need to have an ability to screen these organoids, drugs in a rapid way so that we can say, hey, is the disease there or not post-drug treatment? And so this is really an important point. So I'm going to show you today an example of that, if time permits, as to how we go about doing it. Third, we need quantitative assessments of drug efficacy through these, what I call as endotypes. I'll define them as I go along. So we want to be able to say, hey, if I cure these 5 things, I'm going to cure 80. So if I can define those 5 things or 4 things or whatever number of them, then I have an opportunity to address the disease. So this is what I'm going to tell you about today. So this is our summary view of what I'm talking about. We need model systems, whether they are induced pluripotent stem cell derived neurons or organoids. We need measurements, which will give us quantitative assessments of this data. We need screening approaches, which will allow us to screen efficiently all these different drugs that we look for. I'm going to talk to you about all these 3 things today as I go along. The first thing I'm going to tell you is that one of the really interesting revelations that came from our work was that why is it that people become demented when they get Alzheimer's disease? So the question we ask is, why are people demented when you get AD? In fact, dementia is the very first signal. The cognitive measures are the very first signals that people get when you go to look at neurodegenerative disorders. You go to a physician, you say, you're forgetting things. Although I will not admit that I'm forgetting things, I still definitely am forgetting things. So therefore the question is memory loss, it is inability to make decisions, those kinds of things. How is cognitive measure? Why are we declining in cognitive measure in the brain? It's not that the brain cells are just dying. Why are we doing it? So in the very first work that we did, we showed that dedifferentiation, which means the neurons are no longer neurons, and neuronal repression of neuron-like behavior, which really loses synaptic connections. We lose our synapses, which is why the memory. And I'm going to jump ahead and tell you the reason why the drugs did not work, even if they dissolved the plaques, was because they couldn't restore the synaptic connections. As a consequence, you are not getting any memory gain. In fact, all these drugs I showed you earlier resulted in more memory loss and not memory gain. And so I'm going to show you mechanistically how this happens in a minute. And so dedifferentiation, we published a paper in Science Advances in 2020, which showed that dedifferentiation neuronal repression defined familial Alzheimer's disease. So, and in fact, the work was done largely, this is a tremendous amount of work by Andrew Caldwell in my lab, who is a senior researcher in my lab now, did really a remarkable amount of work. And so let me walk you through, so many of you are not molecular. I don't want to make this a molecular talk, but I want to give you a flavor of what it takes to do these things multi-scale, from the scale of where you measure the things at the cognitive level to the scale where you measure molecular level. So I'm going to walk you through that. So in fact, the mutations, what we did in my lab was we took induce pluripotent stem cells. You can just scrape the skin from somebody like me, and then you can convert it into a stem cell and convert it into a neuron. You think this is magic. It's not magic. It is well established now, and we can show the characteristics of neurons in patients and in human samples. So we took samples from all these different patients, presenilin 1, all these distinct mutations, and presenilin 2 and APP, transformed these into neurons. And so we asked the question, how different are these neurons from a non-demented control neuron? A person who's 20 years old or a person who's 85 years old and has excellent memory, excellent everything. How does these neurons compare with the neurons of such a person? So I'm going to show you some very intriguing results we got from that. So the way we did this was, there's a well-established protocol. This is not developed in my lab, but other people have developed it. So you can take a stem cell, fibroblast cell, skin cell, just scrape the skin. You can convert it into a pluripotent stem cell. You can convert it into a neuron. And then once you convert to a neuron, you can do all kinds of things to the neuron. You can characterize it from physiology. You can characterize it from molecular perspective, etc. So in fact, the first thing we did was, some of you may be familiar with this. In fact, today is the future of all of these research lies in going all the way from the highest scales to the lowest scales. So I'm going to tell you that omics has changed the landscape of things. And I'll go back from the omics back into higher level understanding of cognition and so forth. And so we do all kinds of sequencing, chip sequencing, RNA sequencing, ATAC sequencing. So my laboratory is now involved in all these multiple sequencing methods. What do we learn? I'm going to give you a summary view of what we learned. I told you that when you do this workflow, change these neurons, stem cells into neurons, and do all these things and integrate, 4 things stand out. And these 4 things are really the hallmark of Alzheimer's disease. Number 1, the neurons are no longer neurons. They become non-neuron-like things, which means they lose their ability to function like neurons. Why? I'm going to tell you before the end of the talk why this happens. And second, the neurons these cells enter into a cell cycle. Many of you know, even if you're not a molecular cellular person, the neurons are terminally differentiated, which means that they don't go through a cell cycle. However, these Alzheimer's neurons start going through a cell cycle. So which really tells us already that they are not functioning like terminally differentiated neurons. Third, neuronal lineage repression, which means they don't lose their neuronal lineage. They don't remember. It's in fact, no pun intended, but they don't remember that they are neurons anymore. So they are going into a non-neuronal state. And then synaptic function loss, very, very important, because cognition is all based on synaptic functions. And so there is no synaptic function loss. In fact, one of the papers that was published much after our study showed through high resolution imaging that there is synaptic loss in the brain you can measure that at the early stages. Okay. There are many, many markers of these. I don't want to walk you through that in great detail. In fact, I'm going to go quickly through the molecular aspects of it to get you a gestalt view of what is happening. So in fact, I'm going to tell you this really tour de force analysis or work. People in my lab, Andrew and others, they took all these multitude of measurements on these different stem cells. I want to only point to 4 things here. One is that we compare all these different patient neurons. You come up with something which is very common. What is common are these things. What is common are these, which is there is an increased pluripotency, which means the cells are no longer behaving like neurons. There is increased cell cycle, and there is some inflammation aspects. There is dedifferentiation, and there is modification at the chromatin level, which I'll tell you about. So therefore, these are the hallmarks of AD. And this is not just formation of plaques, formation of tangles. This clearly is the key difference between earlier perception or not. So now let me go back to inject a point here. If you're only going to address plaques and tangles, you're never going to solve the problem because you're not going to fix the fact that it's no longer a neuron. You want to bring it back into a neuronal state. You want to catch it early so that the synaptic loss is minimized. So this is our entire goal in terms of going forward in Alzheimer's disease. Okay, I hope I'm making sense. So then we decided to do integrative analysis of all these things. We call these things as endotypes, these different molecular features which give rise to a behavioral feature. We call them endotypes. For example, cell cycle, pluripotency, dedifferentiation, neuron specification. So we identified the key, how do these endotypes, how do they create cost? We identified the key molecules which are involved in this. Why is this important? It's important because we can really now do therapeutics using these as starting points, and also ask, will that drug address more than one thing? If the drug is only going to address cell cycle, only going to address pluripotency, it is not sufficient because it will not solve the problem of neurodegeneration. So I'm going to skip the next slide because this is a high-level picture. I mean, people in neuroscience, molecular neuroscience, salivate when they look at this because they see all of the distinct molecules that are involved, who are the regulators. They really enjoy this very much, but I'm going to skip it for this audience. I don't want to go into details. Cell cycle. Let's look at the cell cycle in detail. What is happening? Why is the cell cycle re-entering? So we really asked this question in detail. And with collaborations, we did work on also looking at the proteomic aspects of this as to what changes. Very, very interesting, because what happens in this mutation, in the patient who has a mutation, they go through a mechanism where they introduce a phosphorylation of this CDK6, which really, I mean, not absence of phosphorylation of CDK6, which re-enters the cell cycle, which is what happens in all cells in our body, except terminally differentiated cells. So we go through a cell cycle again, and then these are all the genes. We showed that every one of these genes in the cell cycle, for example, those involved in replication, those involved in damage repair, those involved in checkpoint, those involved in spindle and G2M formation, all of them are activated or increased, telling us that the neuron seems to suddenly, the cell seems to think that it is not a neuron anymore. So it's going back into a precursor-like state. And so the next question that came up to us was, what causes the AD endotypes? Why is it, what is really happening? Why is the cell, what's the cause for the cell going back into a precursor-like state? This is a fundamental question that we are posed. So we said, I had a hypothesis before on this. Hypothesis was that for a cell to alter its state, what should really change is the chromatin. The chromatin has to, because every cell expresses a certain number of genes which are canonical to that cell. So now if the cell changes from one state to another state, that means the chromatin has to undergo a change. So I said the chromatin has to get modified. What causes a chromatin change? To look at this, we looked at the chromatin landscape. We used another technology called ChIP-seq, and we used a technology called ATAC-seq to look at what is open in the chromatin, what is closed in going from a normal neuron into an Alzheimer's neuron. Which genes are expressed in going from a non-demented control neuron into an AD neuron? When we looked at this chromatin landscape, very interesting. This is a technology. The technology is now, I mean, it's, you know, many of you may not appreciate this, but genomic technology has changed completely the landscape of understanding human diseases. Completely change the landscape. So today we can do, for example, I can look at the cell in pathology and ask the question, what's open, what's closed, how is the chromatin shaped? So I'm going to convince you that what we showed was that the chromatin alteration is what is causing this problem, not just in AD, but in Parkinson's disease, Huntington's disease, in ALS, in cancer, in all of those. So I'm going to skip this slide. So we did all of these interesting analysis of these. All I want you to take away from here is the same things that we looked at in the transcriptomic analysis. Same things are changing in the chromatin. What modifies the chromatin is when the chromatin gets modified, it introduces cell cycle. It opens up the regions involved in cell cycle. It closes the regions which are neuron-like. And so as a consequence, those genes are no longer expressed. So let me give you a summary view of this. This is what a chromatin looks like. You know, a chromatin, as you know, a nucleus is only microns, and then it's packed together. A 2-meter long DNA is packed into the nucleus, right? So therefore, it's packed in these things called, you know, I mean, histones, which are packed, I mean, chromatoids, which are packed into histones and so forth. And so it undergoes a change in this topology. The topology of the chromatin undergoes a change, and the topology of the chromatin undergoing change is what causes the cell to change and the neurons to become non-neurons. So I'm going to skip all of these and tell you the summary result. Before I do that, the first thing we did was when we submitted our paper first to high-profile journal, they said, hey, you know, you don't have data on humans. You're doing it in cells. Maybe it is just a cell culture thing. You're putting it into a cell or an organoid. It's not with humans. So he said, okay, great. So we got human data from patient who had mutation in this presenilin 1, very close to mutation that we studied. And then guess what? When we looked at the human data, what's common across is exactly those factors which you looked at in the induced pluripotent stem cell data. We are able to convince the reviewers that the patients show very similar motif, very similar everything. In fact, when you look at the factors that are involved, what's changing is pluripotency, dedifferentiation, cell cycle. This is a head-to-head comparison between the cell-based study, stem cell-based neuron study, and the brain study. And in fact, we show that the same factors are involved. So this is really a functional, comparison between a neuron from a pluripotent stem cell from a patient and from the brain. So the next question that came up was, okay, it's great, but tell us what is happening at the level of a mechanism of chromatin change. So this is what is happening. This is what a normal brain looks like in a normal neuron, in a non-demented control neuron like everybody in this room. And you can see that the regions that are closed in chromatin or things that are precursors, dedifferentiation, cell cycle, they're all closed. They're not open. What's open is neuronal differentiation, which is what happens in normal brain. Let me look at an Alzheimer's brain. In an Alzheimer's brain, everything that is open now gets closed. Everything that is closed now gets open by all these factors, which I won't go into detail. This is all published work. You can read that. As a consequence, what is happening is chromatin alterations alters the state of the cell. The neuron is no longer a neuron. So now you can ask me the question, and everybody wants to ask this question. What is the cause? Why is a neuron ceasing to be? This is all the explanation of what is happening, but what is the cause? So this is a fundamentally interesting question. There are theories across board, all the way from a virus infecting the brain to traumatic brain injury, to your banging your head against a table, to all kinds of things. And so this is really an interesting question. Let's hold on to this till the end of the talk when we come back to it. So here's a landscape picture of what we think is happening. Let me go back here. Sorry. I don't know. Wait for the program to respond.
It's slow sometimes.
Okay. It's functioning like my brain. Definitely, must be. I don't know. I should do a escape or something. Oh, there it is. It's come back. Jack, you're just getting out of the chair has made it happen. You have magical powers. So here's our current view of what is happening. The stem cell, when we are young, in embryonic state, when we are still in embryo form, the stem cell goes into various neuronal cells, so like, epithelial cells, endothelial cells, microglia, terminal neurons, it goes into all these cells. This is the normal case. In Alzheimer's case, it reverses back and goes to this amorphous state where it is neither a neuron nor any of these cells. And then I'm going to tell you that my current hypothesis in our lab, and based on solid evidence, is that it's actually a protective mechanism. If you look at your rest of your body, you have injury, it tries to go back, your cells go back into tissues, go back into a more precursor-like state because they can recover back, except that in the brain case, your synapses are lost, synaptic connections are lost. So your recovery is never going back into a normal brain state. Okay, so that's our current hypothesis, working hypothesis. But let's move on. I'll tell you a little bit more about this. So the next question that came up was, all the things we looked at in this paper was, I'm sorry, guys, I don't know what is happening to my cursor. I'm sorry, good. You're not as powerful as Jack. Oh, there it is. Is it? will come.
Let me have to go back here. Oh, shoot.
Oh, there it is. Okay, okay, we got it back. We got it back. Thanks. I will try to, maybe my finger is too hot, so I'm going to do it slowly. So the next question that came was, what about mutations in other genes that are involved? So Phoebe, who's here, was instrumentally involved in this work, along with Andrew and others. And so we have a paper now, which talks about comparisons for all the other types of mutations that are there. So they did, I won't go into details so much, but just to tell you that it is in a tour de force work using same types of tools and technologies, RNA sequencing, chip sequencing, ATAC sequencing, and extensive integrative analysis by Phoebe, we came to the conclusion, I'm going to skip this slide because all it tells you is the same thing: cell cycle, pluripotency, inflammation, chromatin remodeling, exactly the same things across the different mutations. And in fact, we can compare across different cells, they behave like the same way. So it's telling us that this is not a one-off mechanism in some cases. It is a common underlying theme to, Alzheimer's disease. Okay, I'm going to skip this slide too. We did the same thing with chromatin. We looked at chromatin. One very interesting story from here is, not all of these mutations have the same age of onset, right? They have slightly different ages of onset and different severity. So we did a pseudo-temporal analysis of looking at each of these factors, endotypes. And you can see clearly that those which belong to the same mutation replicates, they come under the same, similar mutations come very close to each other. And presenilin 1 is more severe than presenilin 2, is more severe than APP. It's borne out by the age of onset and the longevity of the patient's life. So we have a clear indication of how severe each mechanism is for each type of mutation. So why is it helpful? It's helpful because when you look at drug treatment, when you look at therapeutics, this is the kind of thing that's helpful to assess the severity and age of onset and the quality of life. So this is our current view of this. This is, you can see, this is stem cell going into a neuron. In fact, different mutations go to different stages. This, for example, the orange one goes here, where you don't go back up to anywhere near the neuron, you go into precursor state. Some of them go close to, this yellow one goes up to a precursor state, presenilin 1. Then you have presenilin 2, etc. So we have a kind of a qualitative view of the severity of age of onset, severity of disease, and the endotypes that we talk about. Okay, now the first question, important question that comes up is, this is all great because we know these mechanisms are good for those patients who have familial mutations. What about those who have no familial mutations? Those who are sporadic, those patients that they get when they're 80 years old. So they don't have presenilin 1 mutation, they don't have presenilin 2 mutations. What happens in those patients? To assess this, are mechanisms similarly shared? Because if it is not shared for sporadic disease, this focus of research is very narrow because it only addresses those patients, 5% of patients who have familial disease. We want really to address the 100% of patients who have Alzheimer's disease. So we then tested, are these similar mechanisms shared in sporadic disease? So Andrew Caldwell took, in fact, nearly 50 patients' brains. We went to the ADRC, which is Alzheimer's Research Center. We got postmortem brains from 50 different patients who had died with Alzheimer's disease. Late onset, sporadic, not late onset, sporadic, which means that there are no mutations, no known mutations in these 3 genes that I talked about. We characterized those different cells, neurons, from these patients. And it turns out that those very interesting 2 things emerge from here. In those sporadic patients, age of onset is not important. It does not discriminate severity. Whether they get it when they are 65, whether they get it when they are 85, it doesn't matter. They get, it mixed all over the place. And the second thing that emerged from the analysis of all the modules was something very interesting, which are the same features that we identified in the familial disease are present in these postmortem brain neurons. They had high inflammation, their dedifferentiation, they lost the ability to be neurons, they had, I mean, the same, exactly same phenomenology was observed. What does this tell us? This is now published in Molecular Brain. And so it tells us that the attacking familial Alzheimer's disease, which is easy to go after, will provide us insights also to sporadic Alzheimer's disease. So there can be similar types of approaches towards drug targeting for these cases. So that's what it tells us. Next question we ask, Gert, how much time do I have? 5 minutes? Okay, I'll try to speed up. So we asked the question, why do these drugs fail? I mean, $3 billion drug, semagacestat. This is called the gamma secretase inhibitor. $3 billion from Eli Lilly. Why did it fail? So we asked the question, what did it do and what did it not do? So I'm going to give you a summary. We took the cells, treated these neurons with these drugs. One is a gamma secretase inhibitor from Eli Lilly. The other is a drug developed by a colleague of mine who unfortunately passed away, Stephen Wagner. And so we used this drug and we used these two drugs to ask the question, let's just ask, what do these drugs do and not do? So when we did this, in fact, I'm going to skip this to tell you that these drugs, I mean, really, one drug kills all of the plaques. They are great in solving plaques. However, when we looked at what they do and what they didn't do, I'm going to give you a summary view of this since I'm short of time. This is the figure I want you to focus on. These drugs do not address all the features. They kill the plaques, but they don't restore neurons, synapses. They don't restore synapses. They don't completely stop the cell cycle. They partially stop the cell cycle. So we now have a screening strategy for doing it. We published a paper, in fact, a screening endotype reversal as a novel strategy for screening in Alzheimer's and dementia last year. So this really provides us with a new opportunity. Now we have collaborations with Johnson & Johnson in terms of using this strategy towards developing next generation therapeutics. Our mechanism shared with, I'm going to stop after this, our mechanism shared with other neurodegenerative disorders. What about Parkinson's and what about Huntington's? This is work ongoing in my lab, almost ready to be published as a first paper. What is Parkinson's? Parkinson's disease comes from all of you know about Parkinson's disease, so alpha-synuclein, which also forms aggregates. And in fact, this leads to also loss of memory, loss of cognition, and so forth. Huntington's disease comes from these repeats called CAG repeats. Again, just like in mutations, if there are more CAGs, CAG repeats in your gene, in one gene, therefore that just forms plaques. And so that's Huntington's disease. What's common across all of these? Michael Fitzgerald Fitz, who's a grad student in my lab, he did analysis of comparison of single cell data and bulk, or I mean, brain data, human brain data across all of these. Guess what is common? I'm going to tell you the summary story here. I'm sorry. What's common is the following. In all of these, even though their origins are different, what's common is chromatin remodeling is increased, cell cycle reentry is increased, neuron differentiation is reduced, and chromatin modeling is again increased, immune response is increased, and non-ectoderm dedifferentiation is increased. Same things you see in the Alzheimer's disease, you're seeing in these other diseases. So we have a manuscript which is currently just about ready to be submitted soon. I've independently shown 5 years ago in collaboration with a Salk scientist on glioblastoma, in the cancer of the brain, you have exactly same mechanisms. These neurons are going back, except that they go into a full cell cycle. They don't stop at a cell cycle entry. So as a consequence, they become cancer. And we published this, years ago, 5 years ago. We didn't understand the implications. Today, we understand the implications. In the very last minute, I think I'm going to stop after this if my slide moves. I'm at the mercy of this laptop. Okay, if it doesn't move, I'll stop, but if it moves, I want to show you just one more thing, quick thing. This is oh, there it is. It's going to move now. Sorry, guys. It'll be seconds. It'll work. There it is, it moved. I tried to do this. What about different brain regions? What does AD do to different brain regions? So in fact, we wanted to look at different brain regions. There are 6 different brain regions. It's a grad student in my lab, Ashai Patel. He decided to look at 6 different brain regions. And then we compared all these brain regions. Many of them show common endotypes and distinct endotypes. It's a work in early progress. In fact, we can see clearly when you look at the enrichment of all these different regions, some of them show dedifferentiation more than others. Some of them show cell cycle reentry more than others. So we have different mechanisms. For example, you can see that there is all these different processes which are common and different between these different brain regions. What is it telling us? It's telling us that in future, when we start doing single cell sequencing in these different brain regions, we'll be able to map how the disease spreads across different regions in the brain. This is our next quest. In fact, we are doing 3 things in Alzheimer's disease. We are growing brain organoids from these patients, from early onset patients. We are looking at single cell data so that we can look at different regions of the brain in more clear mapping. And we want to look at sporadic disease so that we can try to figure out where we can address the future. So I'm sorry that I brought more molecular perspective to you in a whole brain meeting, but this is where I think cognition meets disease at the molecular level. So we really want to understand the fundamental endotype. So thank you for your patience and your attendance.
That was a really fantastic talk. And I love the data-driven aspect of this work. It's really data-driven class. Right? Yeah. Just measure everything, all problems will be solved. And I have a lot of questions, but I will share the questions with people.
Shankar Subramanium, thank you very much for the excellent talk. It was very, very groundbreaking, I would say. For the Alzheimer's disease, it has been thought for a while that they call it type 3 diabetes. So can we? You were saying that we couldn't see this Alzheimer's, maybe detect Alzheimer's with the things in the blood, or maybe some level of sugar, blood sugar, might say at least the tendency to have the Alzheimer's, right, in the near future.
I'm so glad, Yasmin. Thank you for the question. I'm so glad you asked this question. So I forgot to answer a very important question to all of you, somewhat deliberately, which is, what is the cause of Alzheimer's disease? So clearly, the cause of Alzheimer's disease is stress to your cells. Anytime you're stressed to your tissue cells, that stress induces changes in reversing back into your precursor-like state. The stress can be brain injury. It can be serious attack. Diabetes is one form of stress which alters blood pressure, which alters your flow of blood to the brain, alters your metabolic influence on the brain. And so that's why those who have diabetes can have Alzheimer's disease. The converse is not necessarily true. There are a lot of AD patients who don't necessarily have any metabolic disorders associated with it, at least in the beginning stages. So the answer to your question is, the reason why people think so that it's a type 3 diabetes, because many diabetic patients in extreme diabetic state have brain disorders, including Alzheimer's disease. So that's part of the argument. The other minor thing I want to tell you is that the brain has the most number of lipids in the human in human body. And so I don't know how many of you know this. The brain contains the most lipid content. And so as a consequence, metabolic changes associated with lipid dystrophy, lipid changes are brain associated. And that really is one of the other reasons why it is called as type 3 diabetes. So I hope that answered your question.
One more question. Yeah, excellent talk. Thank you.
Thank you.
So I've been recently reading that hearing loss is one of the potential causes for Alzheimer's and a bunch of other things. So my question essentially is, can you connect this with neuroplasticity? Like if somebody is detected with Alzheimer's, sometimes of exercising the brain, the cognition, from a mental perspective, has it been known to improve the quality of life or any such information?
Yeah, I mean, this is again a very good question. So you talked about hearing loss. A colleague of mine who's chair of ophthalmology thinks that glaucoma, for example, vision thing, when extreme case of glaucoma, it's associated with or correlated with AD. All of these are neurons. And so therefore there is plasticity in the neurons. When you change the plasticity in the neurons towards a more non-differentiated or de-differentiated state, you're going to have all these effects. Is one causing the other? Is one correlated to the other? We don't know the answer to that. So one of the projects that we are currently exploring is working with the chair of ophthalmology, where he has got 6,000 retinal, extracts, samples. And so we are working with him now, going to start working with him now on analyzing all those samples to see what is the onset of whether there's a correlation with the physiological phenotypes and whether de-differentiation is a common endotype associated with that. We're just starting to do that.
Okay, thank you very much.
Thank you.